I'm going to tell you a little bit about the work, or some of the work that my lab does, thinking about how petals influence the interaction between plants and the animals that pollinate them. And we're primarily interested in the relationship between plants and their pollinators. So we're primarily interested in this relationship because it underpins a great deal of the species diversity of the flowering plants. So you're probably aware that the flowering plants, the angiosperms, are much more species rich than most of the plant groups that came before, the plant groups that Liam introduced us to this morning, for instance. And a lot of that species richness is driven by the diversity that comes from attracting different animals to carry the pollen and influence the reproductive success of the plants. So we're interested in understanding this relationship because we're interested primarily in understanding diversity. But it's also an important relationship for 21st century plant scientists because, of course, much of the food we eat comes from plants or from crops that are pollinated by animal pollinators. So although a lot of the things we think of as key crops like wheat and rice are not animal pollinated, there are all those statistics out there that tell you that about 1/3 of every mouthful you eat comes from animal pollination. Those numbers are derived in all sorts of interesting ways, but essentially there are a whole range of crops, and I'm showing you some of them here, that are very dependent on animals for pollination. And there are a whole range of crops that are only a little bit dependent on animals for pollination, but because those crops are so economically important, that ends up being a really huge amount of food that comes from animal pollination. So in that last category, I'd put the oilseed crops and the legumes, where depending on the species and the variety, you might only see a 5, 10, 20% increase in yield from animal pollination compared to self-pollination, but because of the amount of land these things are grown across, that results in an enormous amount of food, depending on those animal pollinators. And then there are other crops, and I've got some up here in this corner, that are locally very important to the economies of particular parts of the world, but are hugely dependent on animal pollination. So vanilla orchid here underpins an awful lot of the economy of Madagascar, and it's 100% dependent on animal pollination. So no pollination, no vanilla. And this plant here is very important in the economy of Ghana, and I think in my view has to be the most important crop in the world. It's 100% dependent on animal pollination too. Does anyone know what the flower is? Yeah, it's cocoa. So no insect pollination, no chocolate. Very important for the Ghanaian economy, pretty important for me as well. So understanding this relationship is really useful and practical in helping to underpin some key research into food security. So the relationship between animals and pollinators is studied in my lab using this three-pronged approach. We take traits that attract different animal pollinators and we explore their development. So we try to understand how they're built, what the genetic pathways are that build those traits. We then try and unpick how they work, so what their function is, how they influence the behavior of different sorts of animal pollinators. And I think of these as the how question and the why question. So how is it built, and why is it there? And if I understand how and why, I can then go and look in a phylogenetic context at how that trait changes. So in this example, the trait we're looking at is changing here and here on the phylogenetic tree. If I understand how it's built from this part of the work, I can go and look at the genome here and here and see what's changed. How does that trait change? What's happened in terms of molecular evolution? And if I understand how it functions, I can ask what's changed in the pollinator or the range or the habitat of those species that has allowed that change to occur. So I can build up this understanding of species diversity that comes from understanding this relationship. And we take this approach with all sorts of different floral traits. So we work in the lab on color and shape, symmetry, texture, presence and absence of nectar spurs. But I'm only going to talk today about two traits, and they're two traits of the petal epidermis. So they're both shown in this slide here. This is a scanning electron micrograph of a veronica petal, and you can see two things about the petal cells here on the epidermis that are significant. And I'm going to talk firstly about this patterning on top of each cell. So you see the little folds and wrinkles here. I'm going to start off by talking about those, and then I'm going to move on later in the talk to talk about the total shape of the cells, and these cells are conical or cone-shaped. They're coming out of the screen towards you. So starting with those folds or ridges on top of the petals, on top of the petal cells, if those are arranged in a regular enough way on a flat background, they can generate color. And I'm showing you this with an example from hibiscus. This is Hibiscus trionum. And the flower has 5 petals. They're predominantly white with a pigmented base here. And if you look closely at the flower, I hope you can see that these 3 petals, the pigmented base is purpley red. It's a dense anthocyanin pigment here. But the lower 2 petals, as you look at it, that pigmented area looks blue. And that's an angle-dependent color. If I tilt the flower away from you, these petals up here will look blue, and these ones won't anymore. So the color you're seeing is dependent on the angle with which you're looking at the structure. And when color is angle-dependent, we call it iridescent. So the most frequent place you see iridescence is in something like a soap bubble. All these different colors that you see on the bubble here will change as the bubble floats past you or as you move your head looking at it, and that's because it looks different colors from different angles. And at this point, when I talk about this work, people usually are thinking, I've never noticed flowers being iridescent before. So let me show you a few examples, a few of our favorite examples of iridescent flowers. So here's a tulip variety called Queen of the Night. Just like with the hibiscus, we've got a purple pigmentation from anthocyanin, but I hope you can see the yellow, green, blue. It looks a little bit like oil or petrol on a puddle. That's the iridescent effect that's being caused by those structures on the surface. Here's a daisy. This is an Ursinia species. It's doing a similar thing to my hibiscus. You've got angle-dependent blue at the base of the ray florets, and you can't see it on those ones for the way they're positioned. But actually, a lot of the iridescent flowers we work with don't look iridescent to me. So here's one. This is Grelum humifusum. It's a South African endemic. And to my eye, it just looks intensely metallic, very yellow, very strongly colored, but I don't see any angle-dependent variation in color. And that's because to the human eye, the yellow pigment in this flower is saturating. It saturates out all the other effects, and all you can see is the pigment. I can show you the iridescence by peeling the epidermis off this flower, putting it on a microscope slide, and just varying the angle you're looking at it from. So as I move the angle, you can see it reflects red light, that angle, then yellow, green, blue. So as you change the angle, you see the different colors. But you can't see that on the yellow flower, neither can I. But that doesn't mean that the insects that pollinate these flowers can't, because of course insects have very different visual systems to people. So although we can't always see these effects, we can measure them. Being able to measure them lets us then work out whether it's worth going on and doing further analysis with the insects. So this is just to show you how we measure them. We take reflectance spectra of the flowers. So these are measurements where you shine white light at the flower, you collect the light that comes off, and you see what you're getting. And so this is a sort of standard reflectance spectrum graph. We've got wavelengths of light on the x-axis and amount of light on the y-axis. Purple curve is a flower that I would say was blue, because its peak reflectivity is in the blue part of the spectrum. The yellow curve here is a flower that I would say was yellowy-orange. Its peak reflectivity is in the yellow part of the spectrum. But actually, each of these curves is taken from the same tulip, the one I showed you before, and all we've done is vary the angle here with which we're collecting the light that comes off the petal. So we can see those different colors at different angles. Now, this is a simple way of looking at it, and this is how I prefer to look at it, but it's only showing you data from about 6 different angles. We actually take these measurements every half a degree, and then we present them as a heat map. So you've still got wavelength along the x-axis. Now you've got the angle we're taking the measurement from on the y-axis, and the brighter red it is, the more reflectivity there is. So this particular surface at 10 degrees looks yellow, and at 20 degrees looks blue. Now, in fact, it's the diagonal line there that tells you this is iridescent. If that was a structure that was a single color, you'd have a vertical line. It would be the same color at all angles. And that diagonal line, in this particular example, isn't a real flower at all. That's perfect iridescence. You've got a perfect diagonal. And that's what you see if you measure a perfectly iridescent surface like the back of a CD. If we do this with a flower, we end up with something that looks a bit more like this. So the diagonal line is still there. That's how you know it's iridescent, but it's a lot weaker, and there's all this other scattering going on around it. When we first started working on this iridescent effect, we thought that this was a side effect of working with biological material. It's hard to produce something perfectly regular out of biology. And actually, as I'm going to show you through the talk, this disorder, this less perfection, this imperfection in the iridescence may well be significant in the signaling to pollinators. So the iridescent effect is caused by those ridges I showed you at the beginning, those patterns on the petal cells. And if I show you in close-up on the tulip, you follow them into that iridescent zone. These are the edges of individual cells. So the cells on this petal are very long. They're often up to 100 microns long. And on top of an individual cell, you see these little stripes. Here they are in close-up here. And it's these that are causing the color effect. And they're doing that by acting as what we call a diffraction grating. So if you look at the back of a CD, so this is a compact disc on the scanning electron microscope. This is a good way to make the microscope technician think you've completely lost your mind, I can tell you. These are the data grooves, okay? So that's where your data gets stored. And because they're spaced at about the same frequency as wavelength of light, they act as a diffraction grating. So each of these ridges and troughs interferes with the light as it arrives on the surface of the CD. You get constructive interference in some directions, destructive interference in other directions for the different wavelengths, and the result of that is that if you look at light coming off the CD, you have this full rainbow effect of color. So effectively, you're getting a diffraction effect. If you look at my tulip on the same magnification, on the same scale, you can see that those ridges are at about the same frequency, and they're acting in the same way as a diffraction grating. And if we shine white light on a transparent replica of the tulip, we get colors produced again. But not as good colors as you saw with the CD, and that again is to do with this irregularity. We've got wide gaps and narrow gaps in the CD, in the tulip in cross-section, where if we look at the CD, it would be perfect. So again, I'll come back to that imperfection at the end. So we've been taking our usual three-pronged approach to this problem and trying to understand how you build this diffraction grating, how you build a regular enough structure on the surface of a petal to influence light in a predictable enough way to give a color effect that might influence pollinators. We've been trying to understand how the pollinators respond to that, and then I'll talk a little bit about, at the end, about how it's evolved. So starting with the development. Well, like any other developmental project, if we're going to seriously tackle this, we need a model system to work in. No conventional plant model is iridescent, I'm afraid, so we had to develop our own, but we used the hibiscus that I showed you at the start, and it's actually a pretty good model. It's very easy to grow in the lab. It's actually quite hard to kill. It's got a very rapid life cycle, so it goes from seed to flower in about 6 weeks, and the genome is reasonably small, and the plant is closely related to species that already had sequenced genomes when we started, which of course makes gene discovery much easier. So it's not a bad model system. Now, before I tell you about how we've been using it, I need to tell you what those folds are made of. And they're made of the cuticle on the surface of the petal. And just in case you haven't thought about cuticle for a long time, here's a cross-section of 2 plant epidermal cells. There's the cell membrane, there's the cell wall, and the cuticle is this waxy hydrophobic polymer matrix on the outside that all aerial organs of all plants secrete to prevent themselves from drying out. So this is what protects the plant from having all the water sucked out of it on a sunny day. Now, work in Arabidopsis over the last few years, and also actually in tomato, where the cuticle is really important in fruit ripening and how long the fruit lives on the shelf, has told us quite a lot about how the cuticle is built. So the cutin monomers are synthesized inside the cell, and there's a transcription factor group, a family of transcription factors that activate transcription of the genes encoding the enzymes that make these cutin monomers. They're then transported across the cell membrane through a particular subset of the ABC family transporters, and out here on the cell surface, they're then assembled into the polymer using an enzyme, a polymerase, that's also secreted across the cell membrane and cell wall. So this process is reasonably well understood in Arabidopsis and tomato. And our question really is, well, how do you produce it so that it has this pattern to it and isn't just a smooth surface on top? How do we control the patterning? So when we set out to try and understand this, we thought, well, the first thing we need to do is just look and understand what changes when we start to see these folds appearing on the petal surface. And we figured out quite early on that actually, so long as the cells on the petal epidermis were growing equally in all directions at once, so there's isotropic growth here, the x and y axis of these cells is about the same size, we never saw those ridges appearing. But as soon as the cells transitioned to starting to grow anisotropically, so that they're longer in one axis than the other, then we started to see the folds appearing. And so we started to think that perhaps this folding or ridge pattern is something to do with the stretching of the cells, and perhaps stretching of the cuticle, or compression of the cuticle as the cells stretch. And if you look in the engineering literature, there's actually a whole lot of literature that applies to this problem. So I'm showing you here mechanical buckling with a roll of cling film, or saran wrap. If you pull it, you generate ridges, or buckles, or folds, or wrinkles in the material. And that's to do with the thin film that the cling film consists of, under pressure, generating those sorts of buckling. And so in principle, a mechanical buckling model is actually sufficient to explain what we're seeing in our hibiscus flowers. The idea is that the cuticle is a thin film, it's sitting on top of a stiff substrate, which is the cell wall, and if we apply a sufficient amount of stretch and compression to it, we should be able to generate this sort of buckling pattern that might explain our diffraction grating. And if that's the case, then we only have 2 parameters to deal with here. We have to understand how the cells stretch, so we need to understand the direction and extent and control of cell growth. And we need to understand the thickness of the thin film, because that's an important parameter in the engineering models. So that's the amount of cuticle. How much cuticle there is will affect what happens. So working with colleagues in Manchester and Nottingham, we generated a a mechanical model that in principle explains all of the patterns that we can observe on real plant petal surfaces. And so this model just takes into account those two sets of parameters. So we have these lambda terms here and here, which are the stretch on the cell in the x-axis and the y-axis. And you can see that we generate different patterns, which reflect different patterns of cuticle patterning. And then we also have a beta term, which is at the top here on the graphs, the amount of cuticle, and that changes whether those different equations interact and intersect and generates the different patterns in different places. So the model is very simple, and it was very much a starting model and something to give us something to get our teeth into. To test the model, of course, we want to do what any sensible developmental biologist would do, which is to change the things that are the parameters in the model. So we want to modify the amount of cuticle, the thickness of that thin film, and we want to modify the stretch, the amount of which the cells grow. And then, of course, we want to see what modifying these things does to the patterning of the cuticle, look back at our model and see if it all fits together. So we had to develop a transformation system for hibiscus in order to do this. Fortunately, it's very straightforward. It's an easy system to transform. We just use agrobacterium transfection of hypocotyls, go through a tissue culture process, and in about 3 months, we have plants, mature plants in the greenhouse. So we're able to perturb genes that should perturb those two features that we think are important in our model. And what I'm going to show you is that we're taking a quantitative approach to this. So I should say at the beginning, I don't have a finished answer for you. This is not a talk with this is exactly how it works story. It's a talk with a lot of work in progress. But what we've been doing, because we want to change the thickness of the cuticle in this case, in a quantitative way, we don't just want to get rid of it. I'm pretty sure if we get rid of the cuticle, we won't have ridges in the cuticle. We want to reduce it by 10% and 20% and 30% and so on, is that we're taking a quantitative approach where we make a battery of constructs for each of the things we're working with. So in this case, we're looking at the SHINE gene. That's the transcription factor that controls the beginning of the cuticle production pathway. And we've made a battery of constructs with a constitutive activator that should give us lots of extra cuticle, a straightforward ectopic expression construct for a bit more cuticle, a knockdown for a bit less, and a constitutive repressor for a lot less. And the idea is that we put all of these into our plants, we make a lot of transgenic lines. These things all vary in their expression as well, just because of positional effects in the transformation process. And the end result is a lot of different lines with a lot of differences in the amount of cuticle they produce, all of which we can compare cuticle thickness to the model, to the pattern we get, and see how it all fits together. And so just to show you a couple of data points, for instance, this is a line where we've increased the amount of cuticle using an ectopic expression of that SHINE3 gene, and we're looking here at the back of the petal, which in wild type is smooth, and in the transgenic line is now starting to buckle and produce ridges as we increase the thickness of the cuticle. And that's exactly what the model would predict. So playing around with the amount of cuticle, the thickness of the thin film, does indeed affect the patterning. What about playing around with the growth of the cells, the amount of stretch? Well, that's been a lot harder to achieve. So we started off thinking, well, if we put in a cell membrane-targeted fluorescent protein, we'll be able to track cell growth. That'll help us work out exactly what the lambda terms are in our model. And we can do that fairly easily by simply measuring stretch before growth in the x and y-axis of the cells, and then after the growth, and that calculates those lambda terms. And if we do that and put the hibiscus into our model, it sits here. So the good news about that is it's in the right place. According to our model, it should have regular striations in the long axis of the cell, and it does. The bad news is that it's a very long way from any boundary which would allow us to change the patterning that we would expect to get. And so again, we've been using a transgenic approach and finding life a lot harder. This is us changing the shape of the cells with a gene that we knew would reduce growth in the long axis of the cell, and sure enough, we've reduced the cell length from about 100 microns to less than 40, but as you can see, these short cells still have ridges on them, and when we patch them into our model, we've moved the cell shape to here. So the good news is it fits with the model still. We're in the region where we should have ridges, and we do have ridges. The bad news is that isn't really what we wanted to achieve. So we're currently using a different set of transgenic approaches and combining them with pharmacological approaches using cytoskeletal modifying drugs and hormones on those transgenic lines to try and move cell shape further across some of those boundaries. So we haven't managed to prove or disprove our model yet. We've got data that fits with it. But one of the great things about having a model is that it forces you to think really hard about whether everything that you see can really be explained by the model you have. And as we started to look more closely at our model, we realized that we'd missed something. And the something we'd missed came about because what our model says is that if two cells have exactly the same shape, so the same amount of stretch, and the same thickness of cuticle, they should always have the same pattern of cuticle. And that's absolutely not what we see, because we only see that ridge pattern on the upper epidermis of the hibiscus petal and not on the lower epidermis, the back of the flower, where the pollinator doesn't see it. So if you look closely at the cells in these two regions, they're pretty much identical in shape and stretch. There's no difference in the lambda terms. And we can stain the cuticle. So here's the cuticle stained with a lipid stain here in red. And we can then measure its thickness. And so the thickness of the cuticle on this upper epidermis is the same as the thickness of the cuticle on the lower epidermis. And yet they have very different patterns. You can see the ridging here, and there's no ridging on the smooth lower epidermis. And in fact, it was this set of images that made us think what the thing was we were missing. Because actually, these images, although you see the staining is very different, there's a lot more red stain on the upper epidermis, it's actually the same microscope slide. It's just two images taken of the same slide. And the upper epidermis always takes up more stain than the lower epidermis. And that's because it's a different chemistry. So it's very easy to think of cuticle as just a thing. It's an amorphous hydrophobic polymer matrix of cutin. But actually, there are a lot of things that you can do to make cuticle chemistry different. You can put waxes into it, you can put flavonoids into it, you can put all sorts of other compounds into it. And if you vary the chemistry of your cuticle, that's going to vary the stiffness. And the one other thing that the engineering literature says is very important in mechanical buckling is the stiffness of your thin film. If you have a very stiff thin film, it won't buckle in the same way. If you have a very, very flexible thin film, then it will buckle in a different way, and it won't generate regular ridges. So we needed to find a way of analyzing the chemistry of our cuticle to confirm this idea that it was different. And what people usually do to analyze cuticle chemistry is grind up a tomato or an Arabidopsis leaf and extract everything on the surface. That wasn't going to help us because we wanted to look at different parts of the same petal. And so we worked with colleagues in the chemistry department in Cambridge to develop a technique that we call liquid extraction surface analysis. Essentially, we put a drop of solvent onto the petal at defined places, suck it back up and put it straight into a GCMS, and then we can identify what comes out. And a simplified version of what comes out. So on our petal, those ridges are only on the pigmented region. This region has smooth petal cells, and on the graph here, every one of the white circles is a compound that's unique to the smooth cuticle, and every one of the red diamonds is a compound that's unique to the ridged cuticle. And the take-home message is the cuticle's very different in the smooth part of the petal and the ridged part of the petal. So what we can do in more detail here is now focusing on these compounds and think about which elements of the chemistry might be important in changing stiffness, and again start to play around with those transgenically and see how that affects the patterns we produce. So again, just to show you a couple of data points before I move on to the functional work, here's a transgenic line where we've increased the expression level of a transcriptional repressor of wax production. So the cuticle has less wax in it, less long-chain fatty acids. And you can see that we're perturbing the regularity of the striations, but they're still there. And this is my favorite line. This is an ectopic expressor of the polymerase that puts those cutin monomers together on the surface. So if we make more enzyme and therefore more polymerization and therefore a stiffer cuticle, we lose the ridges completely. So there's just the same amount of cuticle there, but it's now too stiff to buckle in the same way. So I'm hopeful here that by combining the chemistry and the stiffness into our original model, we'll be able to come up with a sort of comprehensive set of rules which define how that cuticle buckles to generate that optical effect you can see. Okay. But if it's an optical effect you can see and an optical effect that pollinators can potentially see, you would predict has a function in pollinator attraction. And so I want to show you the work we've been doing to test that idea. And to test this work, to test the function of any floral trait that we work with, we use what I think is probably the lowest tech setup in the entire of Cambridge. So nothing fancy here. We have a box of bumblebees. They're sold for pollination of tomatoes and strawberries in polytunnels. We have a plywood box with a perspex lid. We have a tube connecting the two that we can gate, so we can control who comes in and out of the tube. And the bees come to us with the queen and the larvae in here. The queen stays in here, lays eggs, the larvae hatch. The workers go in and out of the tube to the flight arena and forage. They bring food back to the nest and feed the larvae, which hatch into new workers. So the colony lives in the box for its entire life, and as far as it's concerned, this is the world. And because the bees have never met a flower when they come to us, and they don't know that that box isn't really the world, we can teach them that anything we like is a flower. And so here's a standard training flower, sterile in tube with the lid painted pink. Eppendorf cap on top with 20 or 30% sucrose solution in. And the bees are perfectly happy with this. They'll come in and out and forage on these flowers and take the sugar solution back to feed the queen and the larvae. Now, while they're out foraging, we mark individuals. I hope you can see there's a blue paint mark here, a red paint mark here. And that lets us see which are good workers and which are bad workers. So we want workers that come in and out a lot and do their job properly, not workers that come out, feed, and then hang around in the corner all day. It's hard to get good data from them. So we mark the foragers, we identify good foragers, and we do our experiments with them. And the setup here, the beauty of this setup is that it gives us very good statistically robust data. So I can say that I want to know what the first 100 choices made by 20 independent bees are, and we can go and collect that data using this setup. Because if you do this sort of experiment in the field, you're never sure whether the bee you're looking at is the same bee you were looking at 5 minutes ago or a different bee. That affects the statistics. And of course, if it rains, you don't get any data at all. So the setup lets us test various aspects of pollinator behavior and how they interact with the different floral traits we work with. And I'm going to show you data just for 2 experiments here that we've done with this iridescent trait. So the first is a test of foraging efficiency, and the idea here is to get an understanding of how visible a trait makes a flower to a bee. So with some of those iridescent flowers, I can see it. With some of them, I can't. But to the bee's eye, does this make the flower stand out more? Does it make the flower more visible? And so the experimental setup is that we have 3 identical artificial flowers in our flight arena in an equilateral triangle, and they've each got a sugar solution reward in the center. And all we do is that we time how long it takes the bee from leaving the first flower to finding the second, and from leaving the second flower to finding the third. It doesn't matter which way she goes around the triangle. And we do that experiment many times for one bee, and then we do it for many bees, with all of the flowers, say, blue with an iridescent surface, and then we repeat all that with all of the flowers blue without an iridescent surface, and then I get an idea of the time difference that that iridescent effect has. So here's an example of data. Actually, this is done with red artificial flowers. So the 2 bars represent that first flight from flower 1 to flower 2, and the second flight from flower 2 to flower 3. So with plain red flowers, it takes the bee 4 or 5 seconds to find a flower. If I make those red flowers iridescent, and we've done that here by adding a replica of the surface of a CD to the flowers, so they're perfectly iridescent, you can see that it makes them easier to find. The bee finds those flowers in 2 to 3 seconds. And I think that's fairly obvious and intuitive how that works. If I drop an orange button on the floor here, if I'm looking for it, if it's not iridescent, it might take me a while to spot it. If it is, as I move my head looking for it, the color flash, the color change with the viewing angle change of my head will make the button stand out more. And I think that's what's going on here for the bees too. So iridescence makes the flowers easier to see. But given that real flowers aren't quite as apparent to my eye as a CD, given that the iridescence is imperfect, there's all of that disorder and messiness to it, what happens if we do it with a flower? We were worried that perhaps we would end up back up here. And so we made a similar set of artificial flowers, but this time, instead of pressing the surface of a CD into the artificial discs, we pressed the surface of a tulip. So we've got a replica of the surface of one of those iridescent tulips. And actually, quite the contrary, you find that those discs made with a floral iridescence, a disordered, imperfect iridescence, are just as visible, if not more so, as the perfectly iridescent flowers. So that disorder is not making that flower less visible to the bee. It's just as visible as a perfectly iridescent flower would be. Okay, so maybe that explains why disorder is tolerated in the system. There's another experiment that we wanted to do, and that's about how bees remember flowers. We've shown you that iridescence makes them easy to see. What about learning how to remember them? So if you watch a bee out foraging, she doesn't go to a blue flower and then a yellow flower and then a red flower. As soon as she finds a rewarding flower, let's say it's a lavender, she'll stick to lavender for the rest of that foraging bout. And that's because she forms what we call a search image. The way the insect brain works, it's much easier, rather than making a decision each time you're faced with multiple flowers, just to form an image of what's good and stick to that image. So in principle, iridescence might be a problem for search image formation. It might be a problem for this way of thinking, because actually, if the thing changes color every time you change your head, it's going to be quite hard to remember what it should look like. So the idea of this experiment is that we let the bee forage for 15 or 20 foraging bouts on blue flowers, all of which have sugar in. So she should learn that blue is good. She should form a strong search image of blue. We then replace a couple of the flowers with what we call distractors. So these are slightly different color from blue. They're purple. She can see the difference, but it's quite hard. It's not like blue and red. They're quite different to the bee eye. These are quite similar. And if she's formed a good search image of blue, she should ignore these flowers now. She should ignore our distractors and continue foraging on blue. Again, I can repeat that experiment multiple times, multiple bees, with none of the flowers iridescent, and with all of the flowers with an iridescent surface on top, and see how the iridescence affects her ability to learn. So here's the data again for non-iridescent flowers and perfectly iridescent flowers with a CD surface on top. And we're looking here at choices out of 20. The bee gets it right about 70% of the time on the non-iridescent flowers. So that's her optimum search image formation in this test. She can only manage 70% accurate. If I make them all iridescent, that goes down to 50-50. It's no better than random. So she can't form a search image of those flowers if they have a perfect replica of a CD on the surface. But if I use the tulip replica, as I showed you with the last experiment, same sorts of results for non-iridescent, but the iridescent flowers, she has no problem with search image formation. It's just as good as if there's no iridescence at all. So actually, the floral iridescence is not compromising search image formation in any way. And although we started off thinking that it was an unfortunate side effect of biology that this floral iridescence was a bit disordered and messy, and then with the first experiment I showed you, we thought, well, okay, but maybe it doesn't matter that it's a bit disordered and messy. I think what we're learning here is that it does matter, and actually it's optimal for the visual system of the animal that's receiving it. Now, that being the case, it's perhaps not surprising that iridescence has evolved multiple times across the flowering plants. This is a phylogenetic tree of the angiosperms by order, and you can see in each of the orders that I've marked in red, we've identified iridescent flowers. They're all producing their iridescence using this same diffraction grating effect. But actually, when we start to look at them closely, they're quite different in nanoscale structure. So that's the hibiscus I've been showing you. Here's the grelum. That was that intense metallic yellow flower I showed you earlier. And the diffraction grating here has a much different aspect ratio, so much higher ridges and much narrower spacing between them. And actually, that aspect ratio, according to the engineering literature, cannot be produced by simple mechanical buckling. So we may have convergent evolution here through different mechanisms, through different models, and I think I'm going to have my work cut out over the next few years, understanding the structure of all of these diffraction gratings and working out how they're built. So that will be the evolution part of the story when I give the talk in a few years' time. For now, I thought, since it's a bit unsatisfactory, I might just show you a tiny bit at the end of how we do the evolutionary work with the other feature of those cells that I pointed out at the beginning, which is the gross shape of them. So we've been working on this a lot longer. And this is the conical shape. You can see that these cells are protruding out of the screen towards you. So I'm going to do the development and the function of these in one slide each, so I can tell you about the evolution. The development was worked out a long time ago in Kathy Martin's lab here at the John Innes Centre, and the conical cells here, and this is an unstable transposon-induced mutant line of antirrhinum. The conical cells are produced by the activity of a MIB transcription factor that we call mixter. And it's the master regulator of that pathway, so we know that that's the gene you need to make those conical cells. The function of the cells is primarily about grip on the petal surface. So if you offer bees snapdragon flowers, conical celled wild-type ones and flat-celled mixed mutant ones, this way up, so the bee has to land on a nice horizontal surface and stick its tongue down into this gilson tip to get the sugar, you find no preference for conical-celled flowers. If you offer them the conventional way up, and the bee's got to land on a vertical surface and get its tongue in here, then you now have a preference for conical-celled flowers. And that's not surprising when you look at how bees forage on these flowers in the wild. The claws are gripping into the conical cells to help get a grip on the flower. We can generate similar results in the flight arena setup if we make the flowers move as if it was a windy day, if we dangle them upside down. So any way in which you make them difficult to handle, you increase the preference for that conical-celled form and the increased grip. So we were interested in understanding the evolution of this trait then. We know how you make it, we know what it's for, and we know that not all flowers have it. About 80% of flowering plants have conical cells on their petals. So the first question we wanted to ask was, is it ancient? Is the reason that lots of flowers don't have it actually that it's evolved multiple times and perhaps it hasn't evolved in all lineages? So if we go to the base of the angiosperm phylogenetic tree, so this is the very base of that tree I showed you a minute ago, the earliest diverging angiosperm orders that we still have are the Amberellales, the Nymphales, and the Ostrobaleales. Now, the Amberellales are only represented by a single surviving species, and it's got really weird flowers, so we ignored that. We've been working on the Nymphales here. That's the water lilies, and they have very nice conical cells on their petals. So we chose this one here. This is Kabomba. It's a very tiny water lily, and you can grow it in a goldfish tank in the lab, which makes for easy experiments. And we had a look at how its conical cells are produced. Now, luckily for us, the Kabomba genome only has a single copy of that mixture gene in it. And when we use qPCR to have a look at its expression, it's expressed in those petal organs very early in development, just before the conical cells start to form. So that's the perfect timing that we would expect if it was involved in making those conical cells. We can't do transgenic work in Kabomba, but what we can do is take that gene and express it in a different system. So this is a heterologous expression system. We've expressed it in the tobacco ovary, which normally has flat cells, and you can see it induces conical cell formation and trichome growth. So the protein is certainly capable of generating cell outgrowth. And so that makes us think that actually this pathway, this mixture-driven pathway to produce conical petal epidermal cells, is ancient in the flowering plants. And if it's ancient in the flowering plants and it provides really good grip for pollinators, why don't all flowers have conical petal cells then? Well, part of the answer, I think, is to do with a change in pollinator type. So some of the examples we've been looking at include, for instance, within Nicotiana, where we find loss of conical petal cells associated with a shift to moth pollination. It makes sense, a moth tends to hover, not land, and so that grip won't be so important. And Gabriella Doria's got a poster about that if you want to go and see the work we're doing in Nicotiana. Again, in the Antirrhini, so this is a close relative of Antirrhinum, rhodochiton, it's shifted to hummingbird pollination, and again, it's lost the conical cells. Another hovering pollinator, not a landing pollinator, and so perhaps grip doesn't matter. But I just wanted to finish by showing you that actually you can generate an awful lot of floral diversity, not just by changing type of pollinator, but by changing the way pollinator interacts with the flower. So this is work we've been doing in Solanum, big genus of about 1,500 species, defined by these big enlarged anthers, which dehisce not in a normal fashion, but by these little pores at the tip. And these flowers produce no nectar as a reward. They're pollinated by what we call buzz pollination. The bee comes along, grasps the anthers, vibrates them, and the pollen flies out and lands on its body. And it takes the pollen back and feeds it to the larvae. And within this genus, we see repeated loss of conical petal epidermal cells. They're ancestral to the genus, we can see that, but they're lost repeatedly. So this is a condensed phylogeny of those 1,500 or so species, and each of the arrows is an independent loss of the conical cell form. And I'm just going to focus up here in the Leptostomonum clade to show you how we calculate these as independent. So here's a more detailed phylogeny, a group of species, and you can see that each one of the white boxes here is a species that's got conical petal epidermal cells. Each of the black boxes is a species that's got flat petal epidermal cells. And so I can be fairly confident that Solanum capsicoides down here is an independent loss of the conical cell form compared to, say, this group up here, which have probably lost it somewhere along the backbone here. So if we take, then, pairwise comparisons across the genus, we can look at how these repeated, convergent, independent losses of conical cells are happening. So here's a couple of examples. This is Solanum capsicoides that I just pointed out to you with its flat cells, and here's Solanum cimbrifolia, which is a relative with conical cells. And then in a completely different part of the phylogenetic tree, not in that clade at all, this is Solanum aviculare with flat petal cells, Solanum laciniatum with conical petal cells. These are the kangaroo apples. These are an Australian group. And in each of these two independent losses of conical cells, we've been able to look in at the mixture genes and ask, well, what's the molecular basis of this change in epidermal cell shape? And to cut a long story short, this is qPCR data with the flat-celled species in dark color and its conical celled relative in pale. And you can see if you look at the young bud when those conical cells should be developing, in the flat-celled species, we're seeing much reduced expression of the mixed-alike genes. So we're seeing repeated loss of this conical cell form through repeated change in the expression and the regulation of the master regulator of the pathway. And of course, we need to go in now and look at what's changing the expression pattern of that regulatory gene. But I also wanted to show you how we look at the function in this case. So we designed, I think, what's one of the most complicated bee experiments we've ever done in the lab. It took quite a lot of putting together. So here's an artificial Solanum flower with 5 petals. And each of those petals has been made on a replica of a real petal, either to have conical cells or to have flat petal cells. So this one's got conical petal cells. And we color-coded them slightly differently, so we knew which was which. We've got a hole in the middle of the flower, and there's a real Solanum cimbrifolium flower tucked in the back here with its anther cone and its stigma, you can see there, poking out in the middle, because we wanted to force the bees to forage properly for pollen the way they would in the wild. And so we made 5 of these conical cell flowers, 5 of the flat cell flowers, set them up in the flight arena, and I asked my student to go away and find out what the first 100 choices of 20 independent bees was, so we could find out whether they preferred conical or flat cell flowers when they're foraging for pollen. And she came back to me 3 hours later and said, I'm not doing this experiment, it's stupid. Watch what the bee happens, she said. Oops, means I need to work the technology. So if you watch the bee foraging, the problem is that she doesn't touch the petals. All of the focus of the pollination system, because it's a buzz pollination system, is on the anther cone. So she comes into the flower, she lands on the anther, she feeds from the anther, she grips hold of the anther, she vibrates the anthers, and apart from the occasional slipped foot, she never touches the petals at all. You can see the claws actually on the feet there connecting into the anther cone. So in fact, the answer to the evolutionary question here about why conical petal cells are lost so repeatedly in Solanum is that although we're using the same pollinator as the ancestors of this genus used, because it's interacting with the flower in a different way, you don't need the same trait in the same way, and the trait can be lost either through genetic drift or if there's a selective advantage in losing it, perhaps because there's a cost to making conical cells, that might also explain its repeated loss. It does beg the question, though, as to whether those anthers might start to evolve systems that enable pollinator grip. And if you're interested in that question, then Gwen Davis has also got a poster showing you our work on Solanum anthers and how they affect the pollinator interaction in that system. So I kind of finished by showing you how really badly the PI can design an experiment if she tries. But actually, I think the data we got out of it was very informative, even if the experimental design took about 10 times as long as the experiment did. I also hope I've shown you a little bit about how we take this integrated approach, trying to look at things from different angles. That's very important to us in trying to understand how these traits evolve. And of course, I need to finish by thanking the people in my group who've done the work. I've particularly talked today about Edwige Moirud's work on the hibiscus system, Gwen Davis's work on Solanum, Matthew Dorling and Jonathan Patrick, who do the bee experiments in the lab. And of course, it's all done with a lot of collaborators and support from various funding agencies. But most of all, thank you for staying awake for the last talk of the day.